Fungal Genetics. Silent features of fungi. Vegetative body that is represented by filament called hypha, and it is heterotrophs. Cell wall chitin, often called fungal cellulose, asexual reproduction by sporangiosperms and conidia, sexual reproduction by antheridia and oogonia. Food is stored in the form of glycogen. Enjoy extracellular digestion of food material. General characteristics of fungi. Somatic body of fungus is thallus. Loosely aggregated somatic hyphae is mycelium. Simple spore septum. Uh, sub, Septations is aseptate or septate. Dolipore septum. Organs for absorption of nutrition is ostoria. Mode of nutrition. There are four types of mode of nutrition: obligate saprophytes, obligate parasites, facultative parasite, and facultative saprophytes. In obligative saprophyte, example is Isopus. In obligative parasite, example is rust and smuts. Facultative parasite, Botrytis. Facultative saprophyte is Taphrina. Nutrition of biotops. Compounds are extracted through osteria. Nutrients are soluble and organic. Suppresses senses. Extracellular degradation for cell penetration. Extracellular fat is established and maintained for compatible infection. Fungal cells are useful in the studies of rare events such as mutations and recombinations. The concepts and techniques of fungal, asexual and parasexual genetics has been applied to genetics manipulations of cultured cells of higher eukaryotes, that is, in humans and green plants. The fact that each enzyme is coded by one specific gene was first recognized in fungi and was a paramount importance because it showed how the many cells reactions that take place in living cells could be controlled by genetics apparatus. The discovery arose from biochemical studies of nutritional mutation in neurospora. Genetic studies of heredity and variation. Transmission, the passage of traits from one generation to next generation. Population, genetic diversity and change within natural population. Transmissions, genetics. Similar to more familiar mammalian system with bulk of life cycle haploid, genders are mating type cells are biochemically distinct by morphologically identical. Fungal genome. Fungal genome means fungal nuclei are predominantly haploid, that they contain only one set of chromosomes. This cat is useful in the study of mutation, which are useful, receive and therefore masculine diploid organism. Mutational dissection is the important technique for the study of biological process, and the use of haploid organism conveniently allow for the immediate expressions of mutant genes. Typical characters of fungal genomes. Saccharomyces cerevisiae is 6 MB, that is 6000 genes. Aspergillus nigricans 13 MB and 12,000 genes. Homo sapiens is 1300 MB is 3,000 genes. Little repetitive DNA is single copy genes. 50-60% of nuclear genome is transmitted to mRNA in S. cerevisiae, 33% is in S. communis, 1% in humans. In indoors, few, often none, small 50 to 200 bp. Most higher fungi are vegetative haploids, one genome copy per nucleus. Alternatives other plants, algae, animals risky of haploid. No backup copy in case of genetic damage from UV or chemical mutagens is tend to be diploid or have short. Advantages of haploidy that in multinucleate cells can expose genome to mutagens. Most mutants are deleterious. Cell for advantage mutation heterokaryotic system. That is, advantage mutation it may be centered in a heterokaryotic system. Phenotype of receives mutations are obvious within the vegetative state without generating homozygous recessives. Lab strains of S services now generally include a mutation which stabilizes the haploid state. Nucleus states. Each fungus can be found in variety of nucleus state. That is, number of nucleus and degrees of duplications can vary throughout the life cycle of fungi. Though most commonly haploid, each fungus may have significant periods in each part of the life cycle. Cell cycle. A haploid organism contains a single set of chromosomes in each nucleus. That is, in a haploid organism, they contain single set of chromosomes in each nucleus. Following plasmogamy in filamentous fungi, the number of nucleus doubles, though this need not necessarily go through karyogamy immediately. The state where two genetically distinct haploid nuclei coexist is one cell is called antikaryote. When the nuclei fuse in karyogamy, and nuclei become diploid. In this cycle, fungi are similar to the most organisms. Homo and heterokaryotes. In homokaryotic refers to multinucleate cells where all nuclei are genetically identical. In heterokaryotic refers to cells where two or more genetically different nucleons they share only one common cytoplasm. It is neither in 1n or 2n. Fungal reproduction. Fungal reproduction is reproduction in fungi can be asexual, sexual or parasexual. In asexual reproduction, it involves mitotic nuclear divisions during the growth of hyphae. The cell divisions or productions of asexual pores like sporangia, spores and conidia. The sporangia spores are non-motile spores produced inside the structures called sporangia. In fungi, such as rhizopus and mucor, these spores are disappeared by wind. Conidia. These are non-motile spores produced singly or in chains. Other type of hyphae branched they are called conidia spores. Such spores are produced in fungi like Aspergillus and Penicillium, such as sexual reproduction. It is based on the meiotic nuclear division, fairly typically of eukaryotics in general. The fungal sexual cycle is essential in the same as higher plants and animals, with the fusion of haploid gamete nuclei, that is karyogamy, to give diploidy and meiosis to restore haploidy. Where most fungi differ from higher eukaryotes is that meiosis follows immediately after karyogamy, so that diploid phase is confined to the meiotic cells. Ascomycetes and basidiomycetes, which are sexually reproducing, the difference in the mode of formation of haploid spores. It contains nuclei, therefore product of single meiosis, remain together in group called tetrad. It permits the study of genetics even occur in the individual meiosis. Sexual reproduction, plasmogamy. They take place in following manners, that is, planogametic copulations, gametangial contract, gametangial copulations, spermatiations, and somatogamy. Second, karyogamy. Third, meiosis. Parasexual cycle. The parasexual cycle is a process peculiar to fungi and single-celled organisms. Is non-sexual mechanism for transferring genetic material without meiosis or development of sexual structures. The parasexual cycle resembles sexual reproduction. In both cases, unlike hyphae may fuse and the nuclei will occupy same cell. That is, a hyphae may fuse and the nucleus will occupy the same cells. The unlike nuclei fuse to form diploid nucleus. In contrast to the sexual cycle, in the parasexual cycle, recombination takes place during mitosis, followed by haploidizations, a type of recombination found in certain heterokaryotic fungi. Genetically distinct haplonuclei fuse in the heterokaryon. The resulting diploid nuclei multiply by mitotic division with some crossing over, and diploid homokaryon develop form a diploid conidium. While the parasexual cycle appears to viable mechanism by which genetic recombinations occur, many mycologists believe that it does not play a role in maintaining genetic diversity in the fungi. They have lost their ability to reproduce sexually. Instead, these have been looked upon laboratory phenomenon. Heterokaryon formation is nature. It's not a common event. The pansexual cycle must also be a rare event. Three different kinds of reproduction occur in fungi, each of which provide opportunities of genetic analysis. That is, sexual reproduction leads to recombination of genes at meiosis. Asexual reproductions usually reproduce the genes set as faithfully. Parasexual reproduction derives from a typically mitotic division of unstable cells that produce haploid cells and other unstable intermediate. Thank you.